So I'm moving to the next talk. So we'll be now coming to the in silico approach of drug repurposing. So Dr. Aiswarya Pawar will be taking you through some of the in silico approaches that been used for COVID-19, one of our own work. So Dr. Aiswarya Pawar is a PhD from National Chemical Laboratory in Pune. And after his PhD, since then, she is with us at Prescience in Silico Private Limited as a senior application scientist. So I welcome Dr. Aiswarya. Please come to the stage and present the work as you like to. Thank you very much.
Thank you, Dr. Sudeep Roy. So I'll just share my screen. So is my screen visible?
Yes, it is visible there.
Sorry, yeah. So I'll be talking about in silico approaches in drug repurposing, which has been highly acknowledged recently. So the traditional approach to drug discovery involves the identification of new chemical entities. So the whole process is in two stages: the early discovery and development. In the early discovery, it involves the identification of the targets and potential candidates which are highly selective to these targets. And this compounds further passed on to the developmental stage, where there is clinical trials being done to validate these compounds and on animals and humans for their safety, their toxicity, and then they're further FDA approved and passed on to market. So basically, as you can see, it is not as easy as I speak about it. So as you can see, the whole process is time consuming and cost intensive. So if there is a failed process at any of these stages, there are hardly any backup plans to it, and it cannot be replaced. So the drug discovery, the whole process navigates between this triad, that is the drug, target, and the disease. So it is interesting to reflect on the numbers of the potential objects within this triangle. So for the target space, there are currently 400 drug targets to which the pharmaceutical industry are targeting. And from this, actually, these have been selected from 19 to 20,000 human genomes. So basically, it is 1 to 2% of human genome is currently a drug target. The number of disease is very difficult to estimate, but at the current scenario, there are about 5,000 treated disease. So basically, there are some kind of treatment to these diseases. In total, there are 14,000 disease, and there are approximately about 7,000 disease, which Dr. Srinivas was referring to. So basically, these occur in very small number of patients. And traditionally, it had been neglected by a pharmaceutical company, but certainly the things are changing. On top, we have the drugs. So here we have approximately 1,500 FDA-approved drugs or FDA-approved drugs. So these are new chemical entities, as I've mentioned, and it is from large chemical space of drug-like molecules. So the size of this chemical space can be used for mining to look at to have a potential drugs. But the magnitude of this chemical space is larger than the disease and the target space. So the conventional drug discovery obeys a unidirectional approach, where one compound or one drug can interact with one particular target in treating one particular therapeutic condition. This process is challenging majorly due to overnight decline in number of new drugs that have been approved by FDA over the years. So this increases the risk of failures. And this process, as I've mentioned earlier, it is time consuming, labor intensive, and also highly expensive. An alternate option approach to the traditional drug An alternate approach to the traditional drug discovery has been the idea of drug repurposing, which is similar to the phenomena of recycling. So where you have a clinically approved drug, which or existing approved drug, which can have a new clinical use to it, or which can treat a new therapeutic condition. So the drugs that have been considered for the drug repurposing fall into any of these categories. And there's another concept known as drug rescue, where you try to find out the stalled drugs or the compounds which are already gone in phase 2 or beyond clinical trials, which are already been tested for their toxicity, and which have less side effects. They are being repurposed or reused for other therapeutic conditions. So similar to the pipeline that we saw earlier, the drug repurposing pipeline already bypasses a couple of phases because they have been already tested earlier. So the time required, so the risk of failure is low, and the time and the cost required is much low compared to the traditional drug discovery. So, but there's another aspect to which drug repurposing always has an issue with, is the regulatory consideration and the intellectual property and the patent. So the regulatory guidelines, these guidelines are largely depend on the history of the drugs that we are looking at for repurposing. So so actually what happens is, so basically if the drug has been repurposed, and if it has a new mechanism of action or new therapeutic indication, it has to go through the clinical trials yet again for the FDA approval. Now, another aspect is the patent application and intellectual property rights. So there is no clear indication of the IP protection for the drug that has been repurposed, because the drug target association, or this particular mechanism that you're studying for drug repurposing, might have been already reported in some of the publication or in some online database. So it is difficult to seek an IP protection for the same. So these are some of the reasons that actually hinders the marketing of these drugs that have been repurposed. So biologically, drug repurposing can be of two kinds. That is, when we know the drug and the target, it can address a new therapeutic condition. So basically, the disease that it is treating will be different. And there's another approach, which is an off-target repurposing, where you find a novel target for the same drug, which will have a new pharmacological mechanism. So basically, both the target and the indications are new. Such concepts will help us in exploring the disease mechanism and target space, which has been extensively. So these are some of the success stories of drug repurposing over the years. So the drug thalidomide, launched in 90s by German pharmaceutical company Grünenthal, was used for treating for morning sickness in pregnant women. Due to its adverse effect on the babies and leading to their lethality, the drug was stopped, and again in 2006 the drug was reapproved by FDA for treating multiple myeloma by a different company. So SARS-CoV-2, as we all know, we have been in this evolution of SARS-CoV-2, the COVID disease that has been present past 2 years. So it's a respiratory disease that has infected more than 200 million people and has led to more than 4 million deaths. So after the first outbreak, the virus has mutated into more infectious variants, triggering multiple pandemic waves. The typical life cycle of a coronavirus involves 3 stages. So the attachment entry, replication and synthesis, the virus assembly and release. So onset of infection. So after the entry of the virus, it takes 2 to 3 days for the virus to replicate. And within 3 to 5 days, the virus attacks the immune system, host immune host immune system, and eventually causes severe respiratory symptoms within a week's time. So drug repurposing has been extensively used for this urgent treatment of the viral infection. So there are two ways the drug repurposing has been implemented to have a possible treatment for this virus. One is by blocking the viral replication, which is an early stage of infection. So basically, it is by targeting the RNA-dependent RNA polymerase. So the drug that have been effectively been used for targeting this particular protein is remdesivir, as we all might have known past 1 year. So this remdesivir has been initially being used for Ebola virus, which which was less effective, and then it was repurposed in the COVID-19. So basically, the remdesivir is efficient in treating people those who have been hospitalized with COVID, and has a good recovery rate, or at least it lowered the infection within the 4 days of administration. So this has helped in treating people with infection, but as we know that with the multiple pandemic waves, we are not able to control the rate of transfer of this virus globally. So the second strategy is to consider the host as target for the drug therapy. So this is more effective, and the drugs will be resistant because we don't mutate as quickly as the virus. So we don't have to worry about their resistance. And also, as we know, the virus hijacks the host cell and it uses the host machineries to basically survive and replicate. So there's an interesting interactome study performed by Professor Gordon and a huge group of scientists all over the world. They looked at all possible proteins within the host that interact with the virus for their life cycle mechanism. From this data, they found out 300 protein-protein interaction between the host and the virus. These interaction can form can be a potential target to inhibit the virus. So these were screened against 69 drugs. So these 69 drugs were FDA approved, plus some were also in the clinical trial or pre-clinical trial phases. So our study involved looking at this pool of interaction. So from this pool of interaction, we chose 9 host protein based on their regulatory function, that is if they are helping the virus in the translation or elongation or basically translation transcription factors, if they are helping the virus. And also we need to have a starting structure to start the to have this drug protein interaction. To study the drug protein interaction, we need to have a starting structure. So, and also ligand binding information is also necessary to understand the interaction of the drugs that we are looking at. And from the 69 drugs, we chose 29 drugs which were FDA approved. Rest were in the preclinical trials. So we just only chose the 28 drugs. So similar to the drug or understanding the concept of drug repurposing, we have implemented a fully automated novel drug designing approach into our Prince S3 platform. So basically, this multi-target, multi-ligand module in our platform screens the multi-targets and ligands parallelly. More on this will be covered in the demo in the next talk. So the high throughput screening has been done in 2 stages. First, we dock all the 250 combinations of the host protein and the FDA approved drugs. The stable dock conformers are evaluated using the MD simulations. And based on the interaction energies and the conformational stability, we have chosen only, we filtered out 120 combinations from them and subjected to the second stage where the binding free energies of ligands are computed. So as you can see from the docking study, we see hardly any difference in the docking energies for all the combinations. The interaction energies from the MD simulations did differ. and with large differences basically. So for example, if you look at a translation regulator IF4E2, this one, it has the highest energies for chemostat, but with respect to the other drugs, there is a difference of -20 to -60 kilocal per mole differences. To further quantitate the binding affinity of these drugs, we looked at binding free energies. So in the binding free energy, the highest energies observed for the on target. So on target, which I have already mentioned earlier. So these are the already these target drug combo has been already been there in the literature, or has been already talked about. So the binding free energies for these on target combinations were similar in line to the experimental studies. And we also found out some off target combinations which also showed good binding free energies. So this infers that multiple drugs can bind to same binding site. For example, if you look at HTAG, so there are multiple drugs that are binding to the same binding site with different affinities and different interactions. Some of these drugs that showed a better binding energies from our study are already in the clinical trials in different phases. For example, the drug silmitasertib has already been used for human trials. The second part of our study, we focus on the combinations that are already in the clinical trials, basically the drugs that are already in clinical trials for COVID-19. A drug that binds well or a drug that is efficient should have a stable binding energy and a slow or low dissociation rate. So we observe such a scenario in HTAG valproic acid, nafamostat, and casein silmitasertib. So casein is a kinase which in the recent experiment has shown to have shown to be highly phosphorylated in the COVID viral cells. So in this experiment, when these cells were treated with hundreds of kinase inhibitors, silmitasertib showed a good effective good inhibition of the viral activity. Another interesting fact is of this casein kinase is that it has a direct physical interaction with the N protein. So this N structural protein is part of the coronavirus. So our results highlights the residues involved in binding of the silmitasertib and also the dissociation of the drug from the binding pocket. And it has highlighted that the aspartate 176 and lysine 16 are known to interact with the other drugs and effector proteins from the literature. So this mechanistic behavior gives us an idea of the residues that utilized by this drug, which can be also important in inhibiting of viral N protein. So this drug, as I mentioned, has been recently used in human trials and for treating COVID-19. Apart from this, HTAG valproic acid has also been in clinical trials for the treatment of COVID-19. The interesting fact is that our study from our study, an off-target combination, HTAG nafamostat, showed a good affinity. So this has not been previously discovered or been talked about in any of the literature, and this can be yet another potential combination to look at for the COVID-19 treatment. So in conclusion, the drug repurposing can be used to understand and treat new therapeutics. By using a platform, we can handle large data sets and carry out these computation parallelly. The platform PRIN multi-target multi-ligand helps in predicting drugs with good affinity and high selectivity, which in turn can help us in understanding the drivers in the disease mechanism. Thank you.
Thank you very much, Aiswarya. Fantastic talk. We have learned a lot again in this talk. So continued from Dr. Sinevas, we have seen that how in silico approach can also, pinpoint the drugs which are already FDA approved for other indications. So I'm opening to questions, answer session. So if you have any questions, please type in the chat box. And Aiswarya, currently for you, there is only one question. So Ashish is asking, what is the average error bar in the metadynamics-based free energy calculations?
The average error bar was negligible, will be somewhere around -2 to -5 kilocal per mole.
Okay, thank you. Any other, if not any more questions, so let's thank Dr. Aiswarya again for a nice talk. Thank you, Aiswarya.
Thank you. Thank you, sir.
Thank you. So we are now moving to the last part of this session. So we have learned about repurposing, and we have also seen how in silico approaches are being used for repurposing. And now basically
there's one more question.
Okay.
Look at.
Okay, sure. Yeah, I will look at it. So what are the docking algorithms used here? So these are, as far as I know, because I'm also the author of the paper, I can answer it. So it's a genetic algorithm based algorithm is used for docking using, at the back end of the software, it's a autodock, it runs autodock. Okay. So going to the next part of the